A new dawn breaks, the sun is bright, ideas to share in morning's light. Like seeds we have sown, together we gather, together we have grown. With open minds, let's explore new. Inspiration awaits just for you. Connections to make and wisdom to find. Let's embark on this journey united in mind. So here's to the day, let's embrace the call. Together we rise, together we'll stand tall. A very good morning, everyone. I'm thrilled to see you all to the second day on InCoMAC International Conference on Multidisciplinary Approaches to Chemical Sciences. Your presence is what makes this event truly special, and I hope you all are as excited as I am for what lies ahead. Please join me in welcoming Dr. Atul Goyal, Chief Scientist, CSIR, CDRI, Central Drug Research Institute. We are honored to have you, sir, today. I also extend my warm welcome to Professor Devendra Kumar Singh, Principal, National Postgraduate College, Lucknow, respected faculty members, research scholars, and all my dear friends. I hope you all had a restful evening and are ready for another exciting day of insights and inspiration. It's wonderful to see so many familiar faces as well as some new ones joining us. Thank you for being here. Yesterday was filled with engaging discussions, thought-provoking presentations, and valuable networking opportunities. I hope you find it as enriching as I did. Today, we have another fantastic lineup prepared for you with more sessions that promise to challenge our thinking and expand our horizons. As we move forward, I encourage each of you to fully engage with our speakers and panelists, share your thoughts and ask questions. Your participation is what makes this conference truly special. Let's make the most of today, not in terms of learning, but also in fostering connections and collaborations that extend beyond this event. Thank you once again for being here. Before we dive into the day 2 of the conference, let us invoke the blessings of Goddess Saraswati, the embodiment of knowledge, wisdom, and arts. Her divine presence inspires our learning and creativity. So I ask everyone to join us in offering a heartfelt Saraswati Vandana by Agrima, Muskan, and Unnati. Let's take a moment to reflect on the pursuit of knowledge and the power it brings to our lives.
हे शारदे माँ, हे शारदे माँ, हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। हे शारदे माँ, तू स्वर की देवी है, संगीत तुझसे, हर शब्द तेरा है, हर गीत तुझसे। तू स्वर की देवी है, संगीत तुझसे, हर शब्द तेरा है, हर गीत तुझसे। हम हैं अकेले, हम हैं अधूरे, हम हैं अकेले, हम हैं अधूरे। तेरी शरण हम, हमें प्यार दे माँ। हे शारदे माँ, हे शारदे माँ, हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। हे शारदे माँ।
Thank you all for participating in this beautiful homage. हमारे भारतीय संस्कृति में कहा जाता है अतिथि देवो भव. So as a gesture of respect and appreciation, we'll be giving floral compliments to our esteemed chief guests. And for that, I would request our chief guest to please come up on the stage along with our principal sir to give flower compliments. Flower compliments along with the token of honor to our esteemed chief guest, Dr. Atul Goel, by our principal, Dr. Devendra Kumar Singh. Thank you so much, sir. Please have a seat. Now, I would like to request Dr. Rituraj Mishra, Assistant Professor, National Postgraduate College, Department of Chemistry, for the further proceedings.
Thank you, Ananya. So, I welcome you all to the second day of InCoMAC-2024 conference. And I would like to express my gratitude and would like to welcome our chief guest, Professor Dr. Atul Goel, and we have our principal, Professor Devendra Kumar Singh. We have our head of department of Hindi and also examination controller, Professor Ramkrishna Singh. And we have our one faculty member, Professor Nependra Singh, and our HOD of chemistry, Dr. Vikas Singh. So it is my great pleasure to introduce our plenary speaker for today, Professor Dr. Atul Goel. He has completed his PhD in biomedical and medicinal sciences from CDRI Lucknow and pursued postdoctoral research at prestigious National Institute of Health, USA. With over 25 years of remarkable research experience, he is currently serving as the chief scientist at CSIR CDRI, where he has guided 24 PhD students and 15 postdoctoral fellows. His research focuses on discovery and development of affordable medicines and biomedical devices. Professor Goel's work is highly respected both in India and internationally, with more than 120 peer-reviewed publications and 18 patents to his name. Notably, his team has licensed multiple technologies to industries, including investigational new drugs for bone fracture repair, fluorescent probes for COVID-19 detection, and nucleic acid staining dyes for biomedical applications. Professor Goel has received numerous prestigious awards, such as Vasvik Award 2023, the Nasi Reliance Industry Platinum Jubilee Award 2019, and the CSIR Technology Award in 2019. His pioneering contribution in fluorescent dyes and nanomaterials have earned him international recognition, pushing the boundaries of science and technologies. Today, he will be sharing insights on cutting-edge development in small molecule organic fluorescent dyes and their wide-ranging application in bioimaging and optoelectronic devices. Let us welcome Professor Dr. Atul Goel to deliver this talk. Please welcome him with a big round of applause.
Thank you, Dr. Rituraj, for kind introduction. Very good morning to all of you. And at the outset, I would like to thank Professor Singh, Dr. Rituraj, Dr. Vikas for having me here today on the prestigious conference on multidisciplinary approaches for chemical sciences. Thank you once again. It's really a pleasure for me to be here. And today I'll be talking about the work that we are doing in Central Drug Research Institute for the last 25 years as an independent research. So basically, I'll just touch upon some of the highlights of that because I mean, since it is a kind of technology oriented, maybe most of you would not understand that kind of research that we are doing, but I'll try my best to demonstrate all the research work, the technologies that we are doing at CDRI that will be, that it will reach to you through our slide presentation. So I'll start with the first slide, maybe you can just go ahead. Yeah, so I'm working in the Central Drug Research Institute, which is a premier drug research institute in India as well as in the Asia. And the president of the council, actually, it's fine, I think. Is it okay, or I mean, I can take care of it? Because when you move to the side, then I think. Yeah. So that CDRI belongs to that Council of Scientific and Industrial Research, CSIR, New Delhi. And the president of the council is our Prime Minister, Shri Narendra Modi ji. And the vice president is our cabinet minister, Science and Technology Minister, Dr. Jitendra Singh ji. And our DG, CSIR, is Dr. N. Kalai Selvi ji. And in CDRI, our director is Dr. Radha Rangarajan. She is a renowned professor, and she has done many academic outstanding research work in the area of infectious diseases. So as you know that CDRI is a biomedical research institute focused on the discovery of new drugs related to natural product derived molecules, some synthetic molecules, and as well as the diagnostics in the area of diagnostic to develop some of the disease related technologies that we can develop in CDRI. So the mission is to discover, develop, and deploy therapeutic strategies to alleviate the disease burden. So in CDRI, we have all the research related to starting from designing a molecule till preclinical trials. So under one roof, we have all the facilities, like we have a chemistry department, we have pharmacology, we have pharmaceutics, we have formulation team, we have toxicology, we have molecular biology. So if you talk about any related to biology domain, we have everything. We have one department dedicated, and our scientists are working in different areas. So let's say around maybe 9 or 10 different disease areas that our scientists are working in. So as I just mentioned about the portfolio of the activities at CDRI, in drug discovery and development, we have the new drugs related research work. We have various indigenous technologies, which is very important. Suppose you have a costly medicines. So we try to optimize all those cost-effective routes so that it can reach to the society. We also work on the diagnostics so that at early stage we can diagnose the disease. Like in the case of cancer, if we can diagnose early, we can cure and we can just save our people of the nation. In fundamental research, we have, as I mentioned already, we have a big team of biology group where we have most of the departments we are working on. And in the area of medicinal and process chemistry, which is very close related to the chemistry audience right now here, and I would request and encourage all the students whosoever get opportunity to visit CDRI, please do visit us, so that you will get the feeling that what kind of research that we are doing at CDRI. We also have a human research development team where a lot of doctoral and postdoctoral research students come to our lab for a training, for a PhD degree, and so on and so forth. And also the postgraduate training and skill development program. So all the master's students, you are all invited, you can visit and have a 1 or 2 months training at CDRI so that you see the infrastructure, the technologies, how you can grow up, you can collaborate with us. All the faculty members, they can come to us and then discuss their projects and then get benefit out of it. In the scientific and technical, we have a drug testing laboratory, as Professor Singh just asked about it, that since we have a medicine, so quality is very, very important in that case. So sometimes if you want a testing of that quality, whether it is of good quality, it can be used for human or not, we have a testing facility also so that we can see the quality of that medicine. We do have a GLP test facility. That's a good laboratory practice. So in case of any drug discovery related projects, you have to have all the data in GLP accredited lab. In that GLP, there is no chance of manipulation at all. So whatever you do in your lab, it will be automatically registered in the computer system, and one cannot change any data. So that robustness is very, very important, and trust, honesty is very important when you work in the area of drug discovery, because as you take, this drug will go to the human only, human consumption. So quality matters for everyone. So that's how all the data which is required for making a drug, it's important to have in the GLP accredited lab only. We do have all the biological screening, and we have a very good infrastructure related to all the instruments. Like for chemistry, we have everything. We have several NMRs, mass, IR, HRMS facility, and not only this, we have a UV spectrometer, fluorescence spectrophotometers, and so on and so forth. And anybody can go there and then collaborate, and then they can be benefited using our own facility at CDRI. So as I mentioned that we work on the microbial infection, parasitic infections, viral infections, cancer, metabolic disorders, endocrine disorders, neurodegenerative disorders, and reproductive health. So most of the disease areas that we do cover at CSIR CDRI. And for drug target and mechanistic studies, we also have a very good team of chemoinformatics, computational biology, biochemistry, and structural biology. All the scientists, they help in preparing the mechanism identification of a particular disease area, why this disease is happening, what are the genes involved, which are triggering the disease burden, and so on and so forth. And also, once we have the testing of these facilities, we have to have these in a formulation. So we have a pharmaceutics teams, pharmacokinetics team for bioavailability. We have a pharmacology for testing the biological experiment for in vitro and in vivo experiments. Toxicology, which is very, very important, so that animal facilities, we have a very good facility where we can do the toxicity of the new chemical entities and so on. And further, in collaboration with KGMU, SGPGI, we do some of the clinical research as well. So these are the track record of CSR CDRI, where we, since inception, we have 13 new drugs discovered and developed, 6 diagnostics, 80 processes technologies, 12,000 research publications, more than 450 patents, and we have more than 2,000 PhD students, and training programs 2,000, and several PhD training programs that we do conduct since inception. So these are the products, highlights of it. One is the Saheli. I think you may be knowing this contraceptive pill. We have a RT ether for malaria treatment. We have a Keen Mind, which is a memory enhancer. This is in the market. And very recently, this Centroman, which is a contraceptive pill, this has been taken by the government of India for family planning. And very recently, we have developed indigenous qRT-PCR diagnostic kit for COVID-19, as well as dengue, chikungunya, Zika, and on. We are working right now. And in the indigenous DNA staining dye that we have recently developed, generally people are using imported dyes, which are very, very costly. So as the government mission is that we should be self-reliant. So based on that, for the first time, we have developed our own DNA staining dye, which can cater all the biological labs, not only in India, as well as abroad. So these are the recent technologies, 2023. Uh, uh, One, we talk about this green R, which is a DNA staining gel, and we do have a kit also, where you have an enzyme and this dye. So one can directly use this kit for their life sciences research work. And very recently, we have transferred one technology for the quencher, which is required for TechMan Like Pro for COVID-19 detection kit. So now I'll be just touching upon the work in my lab, what we are doing at CSIR CDRI. So mainly our research group is focused on development of new leads for natural sources for the treatment of osteoporosis and bone-related disorders. Particularly based on our extensive research work of more than 15 years, we could able to identify two primary potential molecules called CDRI 399 and CDRI 1500. So one CDRI 399 is a biodegradable bone implant material for bone regeneration. I'll little bit touch upon one slide, particularly dedicated to 399. And another is CDRI 1500 is a new chemical entities. For the first time from the academic institutions, we could able to reach to the phase 1 clinical trial, from hit to phase 1 clinical trial. And this is a purely synthetic molecule, and all the biological testing has been done in CDRI in GLP mode. And furthermore, we are also working in the area of diversity-oriented synthesis of novel donor acceptor-based fluorescent probes, particularly for developing technologies and diagnostics in the area of, let's say, as I just mentioned, staining DNA dyes, lipid droplet markers, vacuole staining dye, neuronal cell body, and also we are working on chemical sensors as hydrazine, biometals like zinc, calcium, iron, and lead as well. So as I just mentioned about CDRI 399, which is a new medicated bone implant material for bone fracture, basically we got a lead from Butia monosperma, which is a plant called DHAC, where we identified one molecule called medicarpin, and based on that medicarpin, we derivatized several of those compounds, more than 1,500 and 200 compounds, 150 and 200 compounds, and we could get one interesting molecule, 399, which shows bone regeneration property. So as you know, accidental fractures in India are very common. And suppose if a patient had that injury and you have a broken bone, so you go to the doctor and doctor fixes this bone by stainless steel pins, rods to fix that bone so that it can be aligned. So there are several side allergies when you put this metallic implants, particularly this high rigidity, and they are very, very toxic actually. When you have a kind of infection in it, it is really hard to treat those infections. So we came up with the idea that if we can have a biodegradable bone implant material using this magnesium property of osteoconductive, which are osteoconductive in nature, to get the strength of plates. And then if we can put our own osteoinductive molecule to speed up the process of healing, I think we have one solution that we can treat our patient using this biodegradable devices. So based on that, we transfer this technology to a company. They are working in that area so that we can have a biodegradable. So if you have a biodegradable implant, after recovery, that screws will go to this bone and it will take shape of the bone itself. So you need not to go for second surgery to remove that metallic implants. So that's the advantage. And then in 2019, we got a President Award for this technology. Can you just click on that? I'll show it just 1 minute. So this is the way it works actually. Yeah, so another molecule is CDRI 1500, which is a orally active molecule. So as you know, in the word, when you have a fracture, you have to take only the calcium supplements. There is no drug to do the rapid healing of that fractures. So generally people, suppose if you have a multiple fracture, it takes around 30 days. So we have developed a molecule based on animal studies, which can have a 50% fast recovery. So suppose in general it takes 30 days, using our own tablets. In animal, what we found that there is a 50% fast recovery. So the patient can go back to their normal work just in 15 days instead of taking this 30 days. So that effective dose that you can see, I mean, how important it is. And worldwide, there is no drug for fracture repair at present. So we got the same lead, 1500 from the nature. And then only 1 milligram per kg dose we got this efficacy. So you see the tablet only 1 mg. So if you count in animal, it's at 1 mg. If you extrapolate in human, it will be a 10 mg tablet for human. So we transferred this technology to a company called Trica Pharmaceutical, and very soon we are going to start the first human clinical trial in Ahmedabad. So now I'll touch upon the work that we are doing on developing the indigenous fluorescent probes and quenchers for RT-PCR diagnostic kit for COVID-19. So I think this slide is very familiar to all of you. You know the harsh time of that COVID-19 pandemic in 2020. So as you know, the first January 27, 2020, the first case of COVID-19 came in Kerala. And then COVID-19 was declared pandemic by WHO in March 2020. And our Honorable PM, Shri Narendra Modi, imposed a national-wide lockdown on March 25, 2020. And during last 3-4 years, you have seen already the different variants like alpha, beta, gamma, delta, and omicron, the last one, omicron, all these variants of COVID-19. So during that lockdown, actually it was told by the government itself that nobody can go to the work, but only those who are working in the area related to COVID-19, like suppose you want to have a work on diagnostics or therapeutics or some other way, only those are allowed to go to the lab, rest were all in the lockdown. So we thought that it's a good opportunity to contribute to serve our nation using our own fluorescent chemistry. So based on that, our team, during the lockdown, whole lockdown, our team was working in the lab itself. Well, people were on lockdown. So if you see the structure of genome of SARS-CoV-2, the virion is covered by spike protein. I think you might have heard in the newspaper as well. And also the membrane and envelope protein, E proteins. The genomic RNA is surrounded by phosphorylated nucleocapsid N protein inside the phospholipid bilayer, as you can see on the left side. And the primer probe set target for different genes. So I don't know how many of you have already seen the report of your swab sample during that period. So if you clearly see, maybe if you have, you can see it, there will be 2 genes which are crucial for COVID-19. So these are listed here, like one is ORF1AB, N nucleocapsid gene, spike protein, and RDRP. So if you see the old reports, there will be 2 genes reported. If a patient is positive, you will get, suppose RDRP and E gene is positive. That means the patient is COVID positive. So I'll just tell you the brief how we diagnose this COVID-19 detection during that period. So when we started this project in the mid-year of 2020, I think you remember the cost of that COVID-19 detection kit per sample was around 4,500 or 5,000, nearly that. So we were curious to know why this is so expensive. It's a simple swab test and then it taking too much. When we dig out into it, we found that people are using the imported Techman-like probes, which is mentioned here, Techman probe, where you have at 3' end you have a quencher, and at 5' end you have a fluorophore. So far, most of, till today itself, I mean, these are all imported and very costly. So we thought that since our lab is working in fluorescent chemistry, can we contribute and we can develop our own indigenous probe to serve our nation? So with that mind, we could able to develop our indigenous Techman-like probes in our CDRI lab. So the main theme of this is in the quenched form, when you have a quencher and fluorophore, the Techman probe is in the native state in the quenched form. But during the primer probe sequencing and elongation, this primer elongation probe increases and chops up the fluorophore. So based on that fluorophores, we get an indication that whether the patient is positive, COVID positive or not. So I'll tell you a little bit about that, how we can analyze those experiments. So as I just mentioned, you need a oligonucleotide where at 3' end you need a quencher and at 5' end you need a fluorophore. So which is very, very crucial. So in general, as you talk about, in this case, like we have an E gene, N gene, S gene, and RDRP gene. So in that case, if you tag each gene with a different fluorophores, one with the green color, one N gene with the yellow color, and S gene with the orange color, and then RDRP with the red color. So if you have a different color, and suppose in that RT-PCR experiment you get a red color, which is tagged for the S gene, particularly for COVID-19. So you can say that if there is a color of fluorescence color of orange, you will say that it's a gene which is belongs to COVID-19 and the patient is COVID positive. So for that you need at least 2 genes, particularly for COVID, to detect whether it is a COVID positive or not. So based on these technologies, we thought that we can have our own indigenous, because these are all imported dyes. So based on that, we could able to design some of our own dyes, as well as we thought to develop a cost-effective procedure for these commercially available dyes as well. And for that, in the 3' end, we need a quencher. So we thought that all these are commercially is very costly. BHQ1, that is a black hole quencher 1, 2, 3. So we thought that can we have a universal quencher? One quencher can suffice all the quenching properties. So based on that thought, we could able to get one CDRI Q2 and Q3, which can suffice this purpose, and then we filed a patent as well in this. So as I just mentioned, during the lockdown, everybody was on lockdown, but our team was there in the lab, taking at risk, and then they were working to develop all these cost-effective procedures for preparing this FAM dye, Psi3, Texas Red, Psi5, and quencher as well, and the cyber grain. So I'll just briefly talk about the chemistry, since this is a chemistry part. So if you see the structure of FAM phosphoramidite, where you have this type of skeleton, which is linked to the phosphoramidite. So that phosphoramidite group is very important to have linked with this nucleosides. So that's the routine chemistry people are using for oligosynthesis. So for attaching at 5' end, the fluorophore, you need to have a phosphoramidite moiety over there. So based on that, we could able to develop a cost-effective method for commercial dye, and it was 8 times cheaper that we could able to prepare in our lab with high quality, which matches perfectly with this commercial dye. Similarly, we could able to develop another orange color dye related to Psi3. You can see this one is the commercial one, and this is the spectra of that commercial one versus that we could able to synthesize in our lab. Furthermore, this is a red color dye. So all these structures that you can see in the chemistry, And during that lockdown only, whatever facility that we had during the limited resources, we could develop this type of dyes in our lab. Again, this is at high, where you have a emission around 630 or 640 region. This is a Sci5, which is commonly used to tag the genes as well as the RNase piece, which is a standard control. As I just mentioned regarding the black hole quenchers, the left side you see the structure of black hole quencher 1, which works only for the green. To quench the green color, you need a BHQ1, but rest, suppose if you want to have a orange or red color dyes, for that quenching you need a BHQ2. So we thought that can we have a combined one quencher which can work from green to red, so that people should not buy these two different quenchers which are very costly. So based on these structures, which is CDRIQ, as I just mentioned here in this slide, you can see by putting different donor and acceptor functionalities, we modulate the photophysical properties of the molecule. Where the below one, if you see the structure, the UV absorption of that, the dark gray color is the commercial one, versus the above is the CDRI quencher, where you see the green to purple and or red color that can be worked for quenching properties for this quencher. So we developed the economical route. The quenching range was 500 to 750, and we can use these probes for Techman-like chemistry. So once we have this quencher, it has to be on solid support. So we used a CPG for solid support, and then based on that, we could able to do DNA synthesis, particularly like gene. And gene you are talking about, we just talk about. So there is a specific sequence for that, oligonucleotide sequence for that. So we could able to synthesize those genes based on DNA synthesizer. And then once we have to attach this 5' fluorophore on a viral gene called E and RDRP, the second structure that you see, through the click chemistry in copper and ACC, where you get a triazole. So for purity, you have to have the HPLC profile of that. We did the HPLC, dual level probes for UV and FL, and we could purify the oligo for that. And once we have that oligo, we can do the RT-PCR experiment for COVID-19 detection. So as you can see, the COT curves, and the beginning of that curve is CT values. So generally people, you might have seen the CT values in your reports as well. Sometimes if it is a 15, which CT value of 15 indicates that there is a high viral load. And suppose if it is a 34 or 35, one can say that it's a low viral load in that patient. So based on that, one can decide, how they have to take rest and what medication they have to take it. So, to check the efficiency of our probe, we do this low viral load also. So we found that our probe can also detect low viral load as well. So once we have these Techman probes in our lab, so there should be a third-party validation. So we submitted our probe to a government agency, C-Camp Index, where they tested and they benchmark with their own commercial kit. And left side, you see the results from the benchmark, the commercial one versus the InCoMAC, which is a CSR CDRI product, and you can see the COT curves. So initially they did for 15 positive and 15 negative sample, and there was 100% concordance. So we could able to detect, our kit could able to detect the patient, whether it is a COVID positive or not. So then we go for the CDSCO approval so that we can commercialize this kit. And there was, we could able to get 100% sensitivity and 98.8% specificity using our own indigenous kit. So we transferred this technology in the presence of then Minister, Shri Harshvardhan ji. and also the quencher technology with our, in the presence of DG CSIR, Dr. Kalai Selvi. And it was highlighted in several newspapers at that time during this COVID. And during that COVID also, we started this work in when there was a Delta and other variants. After that, it was gone on to the Omicron. So government asks us to develop the Omicron selective so that one can understand whether Omicron patient is having Omicron or Delta. So that based on that only, they can decide. For Delta, it was severe, but in Omicron, it was mild. So for doctor, it was essential to develop the Omicron specific kit as well, so we could able to develop that as well. So we put a name called CDRI OM kit for that, InCoMAC OM. So in another, this is the whole summary of the work for detection of COVID-19. Start from developing a quencher, purifying different dyes, purification, then do the Techman master mixes, and then prepare a kit. So in another project on biomedical related work, we could able to develop indigenous DNA staining dye and their master mixes. So this work we started with a industry partner. I think I would also encourage that whosoever, all the faculties, whosoever is want to start their own project, I think first of all you should find a gap in that area. So in that area, if you get a gap, maybe you in collaboration with the industry partner, you can design your experiments accordingly. So similarly we did, as you know, that DNA, RNA, which are important for transmission of genetic information and for our routine function of life. There are several nucleic acid staining dyes which are all imported and very costly. So these dyes are particularly involved in drug discovery research, various gene marker assays, diagnostics, and biomedical research. All over India, people are using only the imported dyes called cybergreen, which is mentioned here downstairs, down this slide. So as such, dyes are generally non-fluorescent. When it binds to DNA or RNA, through that interaction, it glows. So that's how we could able to detect DNA and RNA of it. So during this, when we did the market analysis of that, the majority of the market was captured by cybergreen. So 78% or 80% market was captured, and which is very, very costly. If you talk about, let's say, 500 microliter of that dye, it costs around 20 to 25,000, which is very, very costly. So we thought that can we have our own indigenous dye, which can scatter, which can serve our biological researchers. So these are the in vitrogen. This is a dyes, imported dyes. You can see the range, and we could able to develop in just 5,000 rupees, the same volume. And we could able to transfer this technology to the company, and this is currently available on government e-marketing site, GEM. So Ms. Sajia was involved from our lab, and then Dr. Shraddha was from the BTPL. She's a director there in Biotech Desk Private Limited. So with the help of industry partner, we could develop this DNA staining dye. And you see the structure, it is very cost effective, stable at room temperature. We filed this patent as well, and then it can binds to all type of nucleic DNA, like genomic DNA, PCR products, plasmids, and RNA as well. You see in the left side, these different quantities of our dyes. It can stain up to 1 microgram range concentration. The second slide is for the commercial dye. The third one is also this commercial dye, and see the results of our own CDRI dye. You can see this brightness is there in that. So again, we transfer this technology to companies so that they can prepare a quality batches and they can market in India. We signed an agreement. We transferred this technology on National Technology Day in 2022. And it was launched in February 2023 in the market. So another important work that we did is a quantum dot related functionally modified fluorescent carbon dot. So what we did, it's a watermelon. Generally people are taking it in summertime. So we took the juice of that. We could able to prepare nano particles of that. So there was a hydrothermal treatment we can do as picturized here. And we can functionalize through surface functionalization by using different amines or alcohols or acids so that we can tune the property of that quantum dots. So as in the left side, you see different chain that we introduced during the synthesis so that on the surface of that functionalization, we can get different type of functionalities. We did this TAM analysis to see the morphology of the crystal, the size. The beauty of this carbon quantum dots is a excitable dependent emission. So as you change the excitation, you will get a shift in the emission. And these are, since it is a biocompatible, economically viable, I think that's the best choice. Generally, people are using chemical-based. So since it's a natural product, watermelon juice only, and we can utilize. Even the peel also that we can use, which we throw in the garbage. So based on that, we could functionalize, and in the top side, if you see, there are multiple of cations. So what we found that it is very selective for lead. So generally, if you see, there is a lead poisoning cases in several places where there is an industry, and then effluents go into the river and all, and then you get this lead toxicity. And then one can detect in biological system. So we developed a process for testing it in blood samples. So this is how the cancer cell lines where we could able to detect this lead using our own quantum dots. So in another carbon quantum dot related work, we used this chiku, that fruit. We took the pulp and then again we did the hydrothermal treatment. You can see this nice blue color fluorescence. So this is due to this carbon quantum dots of chiku. We did this analysis, the surface properties of that using the XRDs and all. And we treated this to show that these quantum dots can be useful in animal imaging as well. So we put our quantum dots through tail vein, and we can see the localization in different organ in mice. And that was pretty stable. The animals were safe. There was no toxicity. So we are just trying to develop the markers or kit so that we can use in disease areas, different disease areas. In another one, we also use this beetroot. Again, the same procedure. So when we use this phosphoric acid, we could able to modify this blue into the green one. So two different colors, and then we could able to see in vivo in C. elegans, which is a soil nematode. On the top right side, you can see we did the surface functionalization, and we could able to see the localization, particularly as you can see in the stomach region. In the heart, lungs, there was no particle, but in the stomach, you can see the fluorescence, and also the intestine, that color is shown here. And rest of the dye removed through the faces. So we published this paper in Journal of Material Chemistry, and this is very cited paper. So overall, as I just mentioned, the fluorescent probes in different diagnostic and biomedical applications. So using this simple lactone chemistry, we could generate aromatic compounds. Just one step at room temperature, And we can different functionalize like R1, R2, R3, and R4, different functionalities we can introduce into it. We put a electron donor and acceptor functionality to tune the photophysical properties, which can be useful both in optoelectronic properties as well as for biomedical application. So based on this work, we could able to develop several kind of organelle-specific dyes for vacuole staining, neuronal cell body. We are trying right now for mitochondria as well. So this type of application that we are doing in our lab. So this is the main slide. I would like to acknowledge all my students who worked in lab whole, sometimes in the night as well, to achieve this project in a timely manner. Because if you have an industry project, the timelines are very, very important. So sometimes they have to work night also, late nights. So generally people see like I'm showing the success, but the whole contribution goes to our students. So you see this hard work, perseverance, dedication, lot of criticism as well sometimes when you don't have failures also, so that you have to overcome and to achieve this kind of success in life. And I would like to thanks once again the organizing committee of InCoMAC for giving me this opportunity to present, to motivate all the students and faculty as well. And once again, I invite all of you to come CDRI to visit and see the experience in the lab that we are working in that area. This is an important slide I always prefer to show. So don't be a parrot in life. Be an eagle. A parrot talks way too much. It talks too much, but can't fly high. But an eagle is silent and has the willpower to touch the sky. Thank you very much. So if you have any question, please feel free. Okay. Good, good.
So my question here,
Where are you from? Where are you from?
I work in Shimla Institution of Events
Okay.
and in collaboration with AIMS New Delhi.
Okay, great.
So one question that I have, I was watching your slides on the fluorescent dyes that you have created for the SARS-CoV-2 testing.
Yeah.
As I am also into SARS-CoV-2 testing.
Okay.
What is the correlation that you have got in your experimental data with the false positive rate? Like with the imported probes that we were using for the detection, there was a lot of false positives. In case when the viremia in total was going down, there was a lot of false positive with the imported ones.
Yeah.
So what was the correlation with the false positive rates you got with the dyes that you have
So
manufactured?
the certificate that I think you missed that one. So there was 100% sensitivity and 98.98% specificity. That is particularly for that only. So the specificity belongs to for false positive only. So we didn't get any false positive in that way. So the kit was so robust in that case.
Alright.
And it was the third party validation.
Yeah. Any students? If not, thank you so much for giving me this opportunity. Thank you very much.
So thank you, Professor Goel. It was totally our pleasure to invite you here for the plenary session. So I think that all of us, especially the young researcher, are leaving the session with a renewed sense of curiosity and motivation to pursue our own scientific endeavors with the same level of passion and commitment. So during the talk, it was really fascinating for me to know that when we were struggling from COVID-19, then the scientists like Professor Goel and their team members were continuously working for us so that we could recover from COVID, SARS-CoV 2019, and then we have different kind of variant of this. So it was really fascinating for me. Also, he has shown that because at that time of COVID, when I was seeing that, okay, the COVID is spreading, and we were really afraid of touching anything because we were thinking maybe the COVID virus will be here. So then I realized that what is the importance of detection. So if you detect those things, if we would have a scanner, so that we scan the things and we know here is a COVID virus, we would not have touched that thing. And then the spread was not that too much. But still, there is a different kind of detection which he has shown, that this Techman probe, this was really good. And then he has also shown us that normally we think that CDRI or CSIR is a government organization, but he has shown that, okay, still if you are a government organization, you have to get the certificate from the government of India if you want to launch your medicine. And the most important thing, which is the tagline of our InCoMAC conference, that chemistry through collaboration, he has shown that you will have to collaborate with industry, with the government, if you want to benefit the society. So thank you very much, Professor Goel. And once again, let him give a big round of applause for sharing his invaluable knowledge and expertise with us today. So now we have a tea. So please join us in a tea session, and then we will again start with the session of oral presentation and invited speaker talks. Thank you.